Hello, my name is Dr. Jeremy Petrovich, Developmental Editor for Current Protocols at Wiley. It is my pleasure to introduce our speakers for today's webinar on the Dxflex, the new 13-color CE IVD flow cytometer for both the routine and clinical research laboratory. First, we have Professor Ulrich Sack, MD of the Universität Leipzig Medical Faculty at the Institute of Clinical Immunology. He serves as the head of the Diagnostic Laboratory of the Institute of Clinical Immunology, Medical Faculty, University of Leipzig, Germany. After his medical study at Leipzig University and a fellowship of the Alexander von Humboldt Society at the Clinical Research Groups of the Max Planck Society in Erlangen, he completed his qualification in clinical immunology. Since 2002, he has been holding a professorship at the Institute of Clinical Immunology at the University of Leipzig, Germany. His main interest is translation of recent developments in immunology into clinical laboratories. Therefore, from 2008 to 2015, he supported the Translational Center for Regenerative Medicine, the TRM, at the University of Leipzig in the function of the research director. Furthermore, he was a group leader and advisor at the Fraunhofer Institute for Cell Therapy and Immunology from 2005 to 2015. He is involved in several networks of scientific experts in the field of clinical immunology and laboratory diagnostics. As a technical assessor, he promotes ISO 15189, 17025, and 17043 accreditation in Germany, Ireland, and Arab countries. He continuously is transferring novel laboratory methods into clinical routine diagnostics. His work focuses on cellular immunology with a strong focus on immune function and flow cytometry. His research is based on interdisciplinary research and on the interaction between the immune system and disease conditions in tumors and chronic diseases. In the fields of immunodiagnostics, psychoneuroimmunology, inflammatory diseases, and immune oncology, he has published around 300 research papers. He has been the chair of the IFCC Working Group Flow Cytometry from 2011 to 2018 and promoted education in recent flow cytometry, offering 16 cytometry courses in Europe, Asia, and South America, covering basic science, immunology, and hematology. The book Cellular Diagnostics, edited by Philip Tarnock and Gregor Roth, has set standards for clinical cytometry within the last 10 years. Our second speaker is Dr. Karsten Lang, Global Product Manager for Beckman Coulter GmbH. He currently presides as the Marketing Manager within Beckman Coulter's Flow Cytometry Business Unit and is responsible for the global product management of the new Dxflex CE IVD flow cytometer. After his biochemical study at the Ruhr University Bochum, he earned his PhD at the Institute of Medical Biochemistry of the Center for Molecular Biology of Inflammation in Munster, Germany. Before joining Beckman Coulter in 2016, President Lang held various sales and marketing positions in life science and diagnostic companies. Thank you both for joining us today to present this webinar entitled Dxflex, the new 13-color CE IVD flow cytometer for both the routine and clinical research laboratory. With that, I hand this webinar over to Dr. Sack.
Yeah, hello everybody. My name is Ulrich Sack, and I'm responsible for a routine lab at Leipzig University doing routine and also research analysis in flow cytometry, and we had the challenge to replace one of our flow cytometers, and I would like to show you what we have done. In relation to this presentation, I declare the following real or perceived conflicts of interest. I have not an employment in a company. I get a research grant from Beckman Coulter. None other research support by companies. I have got honoraria from Becton Dickinson and Beckman Coulter. I have no ownership interests, and I'm a consultant of accreditation bodies. This presentation is sponsored by Beckman Coulter. The content is scientific, non-promotional, and educational. Certain products referred to herein either have specific intended uses which are clearly marked by the manufacturer or have not yet been CE marked and are therefore not currently available for this use and might not become so in the future. The views and opinions expressed in these presentations are solely those of the authors and do not represent the position or policy of Beckman Coulter. Beckman Coulter does not accept any responsibility or liability for the accuracy, content, completeness, legality, or reliability of the information presented. This is to show you the content of the webinar we give. First, I introduce you shortly into the needs of my laboratory. I introduce with my colleague from Beckman Coulter the Dxflex. I will show you the advantages of the Dxflex over the old setup. What have we done in transferring the old workflow to the Dxflex workflow? A short summary, and then you have the opportunity to ask us some questions. As you heard in the introduction, I'm a clinical immunologist. I work here at the Leipzig Institute for Clinical Immunology. Beside my work in the clinical laboratory, I'm a technical assessor for ISO 15189, 17025, and 70043 accreditation. I'm active in a lot of research groups in flow cytometry, and what I find is very important, I supervise graduated students, and one of these graduate students is Annabelle Ernst. She's a veterinary doctor, and she's doing research work in my laboratory, and she contributed to the development of this presentation. Our laboratory is a clinical flow cytometry laboratory, and we serve our university hospital and contribute to clinical studies. We have transplanted patients, patients suffering from immunodeficiencies or suspicious on immunodeficiencies, and we monitor patients undergoing immunotherapies. Our laboratory runs under accreditation. We have a flexible scope. This means we can add additional new methods to the scope of our laboratory without an additional assessment. And this way we offer laboratory tests we developed in our own institute and of course IVD. We also offer functional tests, including cellular functions for immunodeficiencies and functional tests for instance, NK cell functionality. And until now, we use 2 flow cytometers with 8 colors. We are happy with these flow cytometers, but they are very old, and there's now an urgent need for a replacement. And looking for a replacement, we found that there is a flow cytometer representing a novel technology. This is a Dxflex. This is a Dxflex we placed in our laboratory. This is a small machine. There is no compressor under the table. It's a big screen to supervise the machine and to analyze the data. There is an automatic loader. It's very small, and it was no problem to place it in our laboratory for daily work. And I think now I hand over to my colleague.
Thank you very much, Ulrich. My name is Carsten Langer. I am, as said in the introduction, the product manager for the Dxflex within Beckman Coulter. And before we continue with Ulrich Sachs' presentation, I would like to give you a little background information on the system. So, what is the Dxflex? It is essentially the clinical version of our CytoFlex research use only flow cytometer that most of you will probably already be familiar with. So it shares quite a few features with the CytoFlex. It has the same laser engine and the integrated flow cell that help increase excitation efficiency. It has the same wavelength division multiplexer design to ensure data collection efficiency with up to 13 colors. It has the compact design that Ulrich already mentioned. It has an acquisition rate of up to 30,000 events per second with 7 decades data resolution and an intuitive software called CytExpert for Dxflex with a compensation library and graphic tools that help you get the most out of the experiments. So what are some of the key features of the Dxflex? This system is CE marked for clinical use for up to 13 colors. It is offered in 4 different configurations, from 1 laser and 5 colors, then 2 laser 6 colors, 2 lasers 9 colors, and finally 3 laser 13 color configurations. And all of these are field upgradable, so it is basically just a dongle change to unlock new functionality in case you purchased a smaller configuration first and want to update or upgrade later. The autoloader that you can see here on the right side of the system can be ordered with the system or can be purchased later as an accessory. And this autoloader operates with the 32-position MCL carousel that you also know from other Beckman-Coulter flow cytometers, such as the Navios EX. The software can work with up to 25 million events per file, and that is for 13 colors or 15 channels, including forward and sideward scatter. So very high limit of events per file. And the new clinical site expert for the Dxflex software has some additions over what you see in the normal site expert software on CytoFlex. So we have improved the user and account management. You will find experiment and report templates. And probably the most important addition is the addition of panel experiments and flagging of out-of-range results that you can, of course, define. One major benefit of the Dxflex over conventional flow cytometers is the use of avalanche photodiodes, APDs, instead of the conventional photo multiplier tubes. As shown here in the diagrams, in contrast to PMTs, the APDs have a linear response to changing gain settings, meaning that a compensation matrix obtained at one gain setting can be used for experiments at different gain settings through simple recalculation, which the system does for you automatically, of course. This has the potential to simplify your workflow, as you can see here. On the conventional PMT-based cytometer seen on the left, whenever you need to change settings, you have to rerun the full set of single color controls to update the compensation matrix and then rerun the actual experiment with the new settings. In contrast to that, on the Dxflex, the compensation can adjust automatically whenever you need to adjust gains. Therefore, the Dxflex, with its APD technology, really helps to overcome limitations of PMT-based systems that become more obvious in high complexity testing, where the effort required for establishing compensation grows beyond linear. So it is really a great tool to enhance productivity in high color flow testing. It is important to point out that the Dxflex is an addition to our existing portfolio of clinical and clinical research flow cytometers, not a replacement really. So we still have the Aqueous, the first load and go cytometer, which emphasizes standardization through automation, including some sample preparation steps, and therefore is ideally suited for routine, highly standardized, low complexity tests. Then there is, of course, the Navios EX, which especially in the LNL space, so leukemia, lymphoma, can offer you a complete standardized testing solution through an integrated system with ready-made panels like our ClearLab 10-color system. And now, finally, we can offer you the Dxflex, which offers you the latest technology, as outlined before, and is very well suited for non-routine user-defined testing in the high-content field with up to 13 colors. With that, I'm finished with my part and happy to hand over back to Ulrich.
Yeah, thank you very much. And now I would like to give you our first impression about this machine. We found that the Dxflex is a very small, compact and quiet machine. It's very easy to connect it with media in the laboratory, so sheet, waste and power supply are very easy to install. It's very straightforward for the environmental conditions. we found a full network connectivity and a barcode scanner, a carousel loader, and a single tube vortex. We have in this machine 3 lasers with bandpass filters, 13 colors, and as you heard just now from my colleague, APDs instead of PMDs. We found the software very easy to learn, and what was very important for us as a routine lab, a very uncomplicated takeover of old panels. In fact, we could use our established panels without any change in a first step to move it to this new machine. There are numerous multicolor panels available, and what is very important for a routine lab, there is a strong support by Beckman Coulter, and we had contact with very competent trainers explaining us everything what to do. We use this machine to analyze samples in our lab, so we have a daily routine. The daily routine is very easy. We have a startup program with distilled water. It takes around 10 minutes. Then the quality control with quality control beads to control laser and signal value takes around 2 minutes, maybe a bit more, but very, very short. And the daily clean with cleaning agent and distilled water is a 11 minutes procedure. The software for the Dxflex is a CytExpert. This software simulates the Calusa software. It's a very transparent software and easy to handle. You see the plots, the gating strategy, and the basic information we need for our daily business. The software CytExpert has various windows you can see here. There is one for the machine control, file, cytometer settings, and quality control. A dot plots windows with a hierarchy with statistics and gating. A run and record window with auto record events, sample flow rate. A window with samples, tube, library, comp, and carrier information, the combo population window, and information about the carrier. This is everything you need to run the machine in a daily approach. One of the main challenges when you introduce a new flow cytometer is to set up new panel. For new panel experiments, it's very easy to set up a new sample and a new tube. The options are very clear in the bar. There are dot plots, histograms, different gating possibilities. There's a combo population with Boolean gates possible. This will include it in the history. And in diagnostics, we very often use combinations, so we can save panel combinations, for instance, lymphocyte panels. And everything can be saved in the panel library. A very important point for a routine lab is the quality control. The Dxflex offers a complete quality control report that is stored electronically and can be found later on. So everything is under control as required by the regulations for a routine lab. This is the compensation. As you can see here, the compensation data are really impressive. It's a very, very thin peak for all these beads that can be used here with the VersaComp antibodies. And this compensation files can be allocated later on to the protocols you use in this machine. Compensation is done using negative and positive Versa-comp beads. It's very simple to do, so the approach is very easy, and up to 13 different fluorochromes can be used. The software simulates Calusa, and this is Calusa itself. Calusa is a research use only software, and a lot of the postdocs, graduate students, and technicians in my lab are familiar how to use Calusa. And with Calusa, some additional analysis can be done with the results. So, the exchange of data between the software of the Dxflex and the Calusa can be done easily. There are some additional features like radial menus, for instance, and this can be used, for instance, to prepare your data for publications. This is an 8-peak bead analysis. 8 intensities of each fluorochrome can be seen here, and these fluorochromes have to be, these beads have to be separated very clearly. And as you see here, these peaks are very well separated. This is really easy to see that the performance of the electronic in this machine is really superior. Here you find it for other channels. There's nearly the same good performance for all the colors we can apply in this machine. And here the last peaks. This is really a very good separation, and we are really excited about this good quality of analysis. We tried to transfer our old workflow to the Dxflex, and it was really impressive how easy this can be done. We could simply transfer the protocols from our old machine to the new one. And later on, we also tried to introduce a new tube. But first, I would like to show you how we transferred our old panels to the new machine. For instance, we analyzed the distribution of cells in samples with well-known concentration or counts of cells. For instance, here I show you the result for CD4. This is the result of the previous machines. This is the result of the Dxflex, and the correlation between both machines was found to be superior with a very, very good correlation. The same was found for the other cell populations. There was nearly no change in the results between the old and the new setting. Here you can see, for instance, the discrimination of the B cells. The gates you find here is exactly the same what we know from older machines, but the options we can apply here to the analysis will be introduced in future, but for the beginning it was very easy to transfer this B cell panel from the old machine to the new one. T-cell differentiation shown here. You are probably familiar with the differentiation of these subpopulations. It looks similar to the previous setting, and the results are very comparable. Treg analysis, the same. NK cells, no difference to the old setting we had in our laboratory. After transferring old panels from the old machine to the new one, the second question was, of course, how far we could introduce new panels in the new machine that are optimized for this new options of this machine, and we tried to apply first one panel with 11 antibodies and 8 colors to this new machine that can be analyzed in a way that requires a lot of additional mathematical procedures to add and to combine markers to find out a lot of populations. And here I show you the panel composition and the plots. We can analyze with this tube lymphocytes, granulocyte subpopulations, monocytes, TBNK differentiation, T maturation and activation, B maturation, and K maturation and activation, and monocyte subpopulations. All these populations can be found in this one tube, and we not yet apply 13 different colors. We are here restricted to use only 8 colors, but you see the huge potential how we can analyze a lot of different cell populations in one tube. To summarize, accuracy and reliability of the machine is very good. The 8-peak beat analysis shows that the instrument is capable of almost perfect peak separation. The evaluated data are almost always comparable with the Control 2 data, and if it does not fit, it's usually a gating problem. Instruments usually have a very little dead volume, therefore little of the sample is consumed and can be used further. So far, we had hardly any problems concerning the handling, and when a problem occurs, the software usually tells quite clearly what's wrong and how to fix it. About the performance of the avalanche photodiode detectors, about the APDs, it's to say it's at least as sensitive like a CytometerFlex, and we expect to overcome the limitations associated with the PMT-based systems. The applicability of the user interface is very good. After an initial training period, the software is very easy to use. In fact, the colleague handling the machine in our laboratory had not too much experience in flow cytometry. It was a very short time of about 1 week to become familiar with the general handling, and after 1 month she is absolutely perfect in operating this machine. It's very clear how to use the user interface, so there are no too much hidden functions, and for the evaluation of diagnostic samples it's really very good. For more complex panels and for research applications, it's very good to combine the software inside the Dxflex with the options of the Calusa software system. And the automated instrument quality control and the standardization procedures are really perfect. And in combination with a compensation library, it can be used easily to produce high quality results for routine laboratories. The daily quality control with the quality control beads has always worked except for one time. And this one time the service helped us immediately. If a problem occurs, it can usually be fixed with a deep clean function. The instrument standardization is very good. The files can be reanalyzed elsewhere easily, and it's very easy to fix even mistakes or errors of operators with very little experience in this field. A panel library and the compensation library are available to save and to apply the files to your analysis. So finally, we are very happy with this machine. I would like to thank Annabelle for providing material for this presentation. And now we are ready to take your questions. Thank you very much.
Thank you, Dr. Sack and Dr. Lang, for that presentation. Our first question involves implementing this, the Dxflex into the laboratory environment. So when introducing this new cytometer, can I simply transfer my existing panel, or does the user need to establish new panels as well?
In fact, this is the case. So when we introduced the Dxflex in our laboratory, we simply could transfer the pre-existing panels from our previous machine to the Dxflex. Of course, this machine offers a lot of additional options. But when you have to introduce a new machine and new panels, it's really a benefit for the lab director when you can start simply moving the old panels to a new machine and not to start with a parallel validation of all your panels and the introduction of a new machine. So it makes things easier.
Is there a specific interface necessary for LIMS connectivity on this machine?
No. So the Dxflex does offer unidirectional LIMS connectivity by being able to export data automatically, for instance, as a CSV file, and this can then be transferred to the LIMS system by using a middleware solution. Or alternatively, you could use our own Kaluza C software also for analysis of data exported from the site expert and use that software for transfer of data to the LIMS.
So somewhat related to that is, so a user has a data collection in Excel. Can you easily transfer Dxflex data into this existing database?
Exactly. It's basically the same. You can automatically export as a CSV file into Excel.
Can you elaborate on how the Dxflex can be field upgraded if a user purchases the minimum configuration, but later on they discover that they need to add more channels?
Yes. So basically every Dxflex delivered contains all their hardware for up to 13 colors already. So every machine delivered has 3 lasers and all the detectors for up to 13 colors. So basically all you need is a USB dongle change that unlocks new hardware functionality. So basically that can be done by any field service engineer within 1 or 2 minutes.
How is the sensitivity in comparison to existing PMT machines?
In our experience in the laboratories, the sensitivity is superior. We started repeating a lot of experiments where we could not find a really relevant difference between negative and positive signals with this machine, and the resolution is higher, so we get very clear signals. So from the point of view of a user, it's really a wonderful machine for such applications.
Yeah, and if I can maybe add to this, so the Dxflex was validated using our DuraClone B27 assay, so HLA B27, in comparison to the FC500 instrument. Most of the samples were concordant, but for the few samples that were discordant and actually positive on the Dxflex and negative on the FC500, we did discordant resolution using an LDT and found that in all the cases where the LDT data was available, the Dxflex data was correct, hinting at a higher sensitivity of the Dxflex.
In terms of the requirements and for the software, is the software that comes with the Dxflex also CE IVD?
The software. Carsten, you? Okay.
Oh, please go ahead. Please go ahead.
The site expert software belongs to the machine, and it's a CE IVD machine with a CE IVD software. So it's one package, and it works very well. It's a fully CE IVD certified software.
Yeah, and also I would like to point out that we don't only have the CE mark for the instrument, it's really the Dxflex CE IVD system, so it's the instrument together with the software and also all the system reagents, so sheath fluid, QC beads, flow clean, they are all CE.
Great, thank you. For QC tracking, does the instrument only track beads, or does it also track internal process controls as well?
Currently, it only tracks the QC beads. This is something where we got feedback from even from the early evaluation side, and we're looking into expanding this. But currently, it's indeed only QC beads.
Is it also possible to use 96 well plates for the measurements with this instrument?
Yes, indeed, that is possible. So the Dxflex, as you might have seen in the pictures, has the autoloader on the right side of the instruments, which usually takes a carousel. There is an option for a plate adapter for this autoloader. That takes flat bottom U and D plates, no deep well plates, but you have some options for 96 well plates. I have to point out, however, that the plate loader option is RUO status only, so this is not CE marked.
So could you please explain a little bit in further detail how the Dxflex normalizes samples?
Ulrich, would you like to take this?
I'm not sure what you mean with normalizing samples. So the system is very stable and we got the same result every time. I'm not sure what you mean with normalization in this context. Sorry.
No, that's understandable. That's something that we can address with the person who asked. So moving on from that is, go ahead.
But maybe the point is a bit with the Dxflex, it's not necessary to do any additional effort to compare results from different machines. So Dxflex gives a very stable result independent on the machine you are running, and so it's a very good comparability between Dxflex machines in various locations. And this is very good for multi-center studies.
Great, thank you. Which software are you using currently? Did you have to develop your own software for your research?
In the moment we use different software systems dependent on the application for routine lab and CE IVD, and for research, for instance, flow. But for transferring the results from the Dxflex to our research purpose, we transferred the results directly to Calusa, and this is very easy to handle, so no problem.
Can you give us some information about the panel report option? Does this have the same features as FC500 Navios?
The panel report option gives you all the features you need for a routine lab and also for a research lab. The complete gating strategy, the panels, the antibodies, the labels, everything you defined with your panels is shown in the reports if you want.
Do you have any data regarding a comparison between the Dxflex and the Navios EX?
I never have done such a comparison because we don't own a Navios EX. Carsten, do you have data, comparison data?
Nothing that is Beckman internal and is like official validation data. So this is something that we would need to work on together with external labs.
Can biosimilar research be done using this machine?
With this
In theory, I would say yes.
Yes.
Please go ahead.
You can do so, but there are no IVD test kits for biosimilar research. So for research, of course, you have to establish your own panels. And we are doing a lot of research around antibody therapies, and this can be easily be done with this machine.
Exactly, and I just wanted to say that, of course, there is also the CytoFlex RUO flow cytometer, which would be our research platform that can be used for biosimilar research. But of course, the Dxflex has essentially all the functionality that the CytoFlex has, plus a number of software options and features that are required in a clinical environment. So essentially, absolutely, you can use the Dxflex for this kind of research.
Do you have experience with MRD testing on the Dxflex? And is the device fast enough to analyze the large number of events that is required?
Again, this is something that is not officially validated from the Beckman-Coulter side, so I can't comment. Obviously, there are no software limitations with regards to events per file recorded. So the ceiling here is at 25 million events per file across all 13 colors. So from that perspective, it is well suited. The maximum acquisition rate is 30,000 events per second. So I would say from that perspective, it is well suited.
How do you deal with spillover on this instrumentation? Is this frequent with this technology?
I mean, there's always spillover, right? And APD doesn't prevent spillover. I think as we try to explain in the slides, the main difference is probably that the compensation that deals with spillover is way easier to do and less time consuming than with conventional PMT-based flow cytometers. Ulrich, what would you say?
Yeah, we had no special problems with it. In fact, we are really able to run a 13 colors panel in this machine, but a bit spillover every time has to be taken into consideration, and I cannot exclude it.
This question is being directed towards Professor Sack. Have you had a chance to assess performance of 13-color leukemia panels on Dxflex?
Unfortunately, not yet, because our laboratory is an immunology lab, and we have a partner lab. It's in the hematology, and we have a strict separation between the panels we run. No, unfortunately not.
How well is the performance of this machine compared to the older models when performing lymphocyte cross-match for organ transplant patients? Do you have any data concerning that?
This is not a specialty of our laboratory, but in general, the performance of the machine is very good. It's a very quick to handle machine with very short times in preparation and in cleaning after usage. So in our hands, the performance was superior.
Have you tried working on flow DSA XM reagents from 1 lambda on this machine? And how is the sensitivity and accuracy in terms of establishing the MCS range cutoff?
No, we did not run such an approach. Sorry, I have no data about this point.
That's fine. Why is 13 colors always written between brackets?
Because it's only an option. You have not to run also 13 colors, and sometimes it can be better to leave some positions free for better separation, but we can in general run 13 different colors with this machine. We tried it in some experimental panels.
Somebody was asking if you could please explain the APD mechanism in more detail.
Yeah, that would probably take the better part of the Q&A session, if not more. I would like to point you at our webpage where we actually have at least 2 technology videos up that deal with the APD technology that's in the Dxflex for more explanation.
Is there volumetric counting available on the Dxflex?
Yes and no. So it's not CE IVD. It's not validated as part of the clinical study. So it's an RUO feature that is, however, available in the software.
One question from the audience is just basically looking at what the cost of the machine is and what the types of available assays and what per test costing is like.
Yeah, for the actual instrument, there are a number of options available, so it's hard to give one particular price even for a list price. So we have 4 different configurations, and it's with or without autoloader. So the basic configuration, which would be the 5-color without autoloader, starts around, I think it's around 80,000 euros as a list price. But then, of course, it depends on what exact configuration you would like to have, and you know what other machinery or reagents you acquire from us. Regarding the test menu, regarding pricing, that's also very subjective. You know, it depends on a number of factors. Usually, there's like a supply agreement for not just one test, so it's hard to give an exact price here. As for validated assays, the only assay validated right now is the Duraclone B27 kit. And this is, as I said, on that one slide, because right now the Dxflex is meant to be more the LDT machine in the complex, high complexity testing section, whereas we still think that the Navios EX is the mainstay in the more standardized testing setting, where we do have more ready-made assays available.
Thank you. So a user is asking that questioning that they are currently using Navios EX. Will it cause a lot of changes in their already established panels when transferring to a Dxflex?
I don't expect it, except very few exemptions. These panels can be transferred without any change to the Dxflex.
How is the data reproducibility in long runs? So, the testing run of around 6 months, and how does that impact the stability of the performance of the APDs?
In our lab, we had the chance to use the machine for now 7 months, and we have not found any shifts influencing the results in our experiments. So we found it very stable, but we have not longer experience.
Is Dxflex software 21 CFR Part 11 compliant?
It currently is not. So this is a feature that is typically more used, for instance, in biopharma research. And here, typically, this is something where we position our Cytometer RUO platform that is CFR 21 Part 11 compliant.
One question is, how do you decide the gain settings that are optimal for the panels you were using on the Dxflex?
Ulrich, would you like to take
the gain settings um, are typical for photomultiplier tubes, but here we have APD. So I'm not really sure how to apply to this machine. So the settings are done in a different way because it's a fully electronic device and not a photomultiplier tube.
Great. Thank you. Well, so we've reached the end of our time today for questions. I'd like to thank Dr. Professor Ulrich Sack of the Institute of Clinical Immunology at the University of Leipzig and Dr. Carsten Lang of Beckman Coulter GmbH for taking the time to present to us this webinar regarding the new Dxflex CE IVD flow cytometer for routine and clinical research laboratories. Any questions that were not addressed will be answered by email when able. This presentation was sponsored by Beckman Coulter GmbH in partnership with Wiley's Current Protocols and will be available for on-demand viewing on the Current Protocols website at www.currentprotocols.com in the webinar section under resources. Thank you for attending.